Good morning, dear students. Today we will learn about de novo biosynthesis of fatty acids. So, specific learning objectives. At the end of this class, the student shall be able to explain different stages involved in synthesis of palmitic acid by de novo fatty acid synthesis, explain enzyme system of fatty acid synthase complex, recall significance of fatty acid synthase complex, explain fatty acid elongation process, explain desaturation process, that is the synthesis of unsaturated fatty acid from saturated fatty acids, explain regulation of de novo fatty acid synthesis. Okay, so this fatty acid synthesis, de novo fatty acid synthesis occurs in liver, kidney, brain, adipose tissue, and lactating mammary glands. So enzymes are located in the cytosomal fraction of the cell. That is, this de novo fatty acid synthesis occurs in cytoplasm or cytosome. So hence it is called as extra mitochondrial or cytoplasmic fatty acid synthase system. So palmitic acid, so 16 carbon saturated fatty acid is the major fatty acid synthesized by this pathway. So in a previous class we have seen the catabolism of fatty acid, that is beta oxidation of palmitic acid, and in today's class we'll learn about how this palmitic acid is biosynthesized inside our body. Okay. So de novo synthesis of fatty acids. De novo means synthesizing the molecule, a biomolecule, from scratch. Okay. So this the source of carbon atoms for this de novo fatty acid synthesis is acetyl CoA. So NADPH is produced from HMP shunt and Malik enzyme reaction. So this is one of the very important component for biosynthesis of fatty acids, and this provides the source for this NADPH is HMP shunt and Malik enzyme reaction. So we will look into it. What is Malik enzyme reaction? Okay, for every molecule of acetyl CoA delivered to cytoplasm, one molecule of NADPH is formed. Okay, so this energy required for the anabolism is, that is for the biosynthesis of fatty acid, is supplied by ATP. Palmitic acid is the first fatty acid produced during fatty acid synthesis. So this is considered to be the basic fatty acid. So this is the precursor for the synthesis of longer fatty acid. So that is, initially palmitic acid is synthesized by de novo fatty acid synthesis method, and further this palmitic acid can be converted to other fatty acids by fatty acid elongation process, and it can also be converted to unsaturated fatty acids by desaturation process. Palmitic acid is a major body component of animals, and in humans it makes up to 21 to 30% of the human depot of fat, that is fat depot. Okay, and it is the major lipid component of human breast milk. So that is why we have studied the beta oxidation of palmitic acid, and now we are studying, or we are learning, the biosynthesis, de novo biosynthesis of this fatty acid, that is palmitic acid. Okay, so the stages of de novo fatty acid synthesis. So there are 4 stages. So the first is production of acetyl CoA and NADPH in mitochondria. Second one is transfer of acetyl CoA and NADPH from mitochondria to cytoplasm. And the third one is conversion of acetyl CoA to malonyl CoA. And finally, the major part of the pathway is the reactions that occur in fatty acid synthase complex. Fatty acid synthase complex is an enzyme complex which contains 7 different enzymes, and these 7 different enzymes catalyze 7 different reactions in different stages that we will look into in your future slides. Fatty acid synthesis. Okay, so now let us study in detail about each that is about these stages of de novo fatty acid synthesis. Okay. So the stage 1, production of acetyl-CoA and NADPH in mitochondria. So acetyl-CoA is the starting material for de novo synthesis of fatty acid. So it is produced in mitochondria by oxidation of pyruvate fatty acids, that is beta oxidation, degradation of carbon skeleton of certain amino acids and from ketone bodies. Okay. So this NADPH provides the reducing equivalent, and the source of NADPH is, that is, it is produced from HMP shunt and malic enzyme reaction. So for every molecule of acetyl-CoA delivered to cytoplasm, one molecule of NADPH is formed. So this is that is for every molecule of acetyl-CoA that is transferred from mitochondria to cytoplasm. So one molecule of NADPH is formed. So here the malic enzyme reaction occurs. Okay, so that we will look into. So and the source of energy for this production of acetyl-CoA is ATP. So the second stage is transfer of acetyl-CoA and NADPH from mitochondria to cytoplasm. Okay. So here the source of acetyl-CoA is, one of the sources of acetyl-CoA is pyruvate. Okay, so there are other sources like metabolism of amino acid and beta oxidation of fatty acids. So acetyl-CoA is produced. So pyruvate, when it is so by the action of pyruvate dehydrogenase complex, it produces acetyl-CoA, and by the action of pyruvate carboxylase, it produces oxaloacetate. So this oxaloacetate and acetyl-CoA react, and by the action of citrate synthase, they form citrate. Okay. So since acetyl-CoA is not permeable through mitochondrial membrane, so this is converted to citrate in mitochondria, and it is transported to cytoplasm. So in cytoplasm, citrate releases acetyl-CoA. So that is citrate transfers the acetyl-CoA. So and itself gets converted back to oxaloacetate. So this process requires one molecule of ATP, and this oxaloacetate is further, by the action of malate dehydrogenase enzyme, is converted to malate. So here NADH2 contributes to reducing equivalents for the conversion of oxaloacetate to malate. So NADH2 is converted to NAD. So malate is converted to pyruvate by the action of malic enzyme. So this reaction provides one molecule of NADPH required for de novo fatty acid synthesis. Okay, so here malate undergoes decarboxylation reaction and it produces carbon dioxide. This is carbon dioxide also acts as one of the component of fatty acid. So NADPH, one more source of NADPH is HMP shunt pathway. So from pyruvate is also obtained from glucose metabolism, and this pyruvate is transported back to mitochondrial matrix, where it can be further again reused for the transport of acetyl-CoA into the cytoplasm. So here in stage 2, mitochondria is not permeable to acetyl-CoA. An alternate or bypass arrangement is made for the transfer of acetyl-CoA to cytoplasm. That is, acetyl-CoA condenses with oxaloacetate in mitochondria to form citrate. So citrate is freely transported to cytoplasm by tricarboxylic acid transporter, and in cytoplasm it is broken down by ATP and citrate lyase to liberate acetyl-CoA and oxaloacetate. Oxaloacetate is, so acetyl-CoA enters fatty acid synthesis, next stage of fatty acid synthesis, and oxaloacetate in the cytoplasm is converted to malate, and malic enzyme converts malate to pyruvate. So during this conversion, one molecule of NADH2 is formed, which is utilized in fatty acid synthesis. That is one molecule of NADPH is formed, So NADPH, so during malic enzyme reaction, NADPH and carbon dioxide are generated, so which are utilized in fatty acid synthesis. So this is about stage 2. In stage 3, acetyl-CoA is converted to malonyl-CoA. Okay. Acetyl-CoA is carboxylated, so that is, so here one carbon dioxide is added to the methyl group. Okay. So when you add carbon dioxide, it gets converted to malonyl-CoA. So this reaction requires one molecule of carbon dioxide and an ATP energy is required, and for the carbon dioxide fixation you require biotin, and this reaction is catalyzed by acetyl-CoA carboxylase. Okay, so that is, so this is carboxylated to malonyl-CoA by the enzyme acetyl-CoA carboxylase. So this requires ATP and requires a biotin as cofactor for carbon dioxide fixation, and acetyl-CoA carboxylase. So this is a regulatory enzyme, that is, this is a rate limiting step. So this step controls the rate of fatty acid synthesis. Okay. So the next thing is reactions of fatty acid synthase complex. So before we discuss about the reactions, we will look into this multi-enzyme complex. Okay, fatty acid synthase is an multifunctional enzyme complex. Okay, so it is an dimer with two identical units. It is having two identical monomers. So here you can see. So this is one monomer, and so this is other monomer. Okay. So the next thing is each monomer possesses the activities of 7 different enzymes and an acyl carrier protein bound to 4-phosphopantothiene. Okay, so here each monomer possesses activity of 7 different enzymes. So here you have 7 different enzymes: ketoacyl synthase, acyl transcyclase, malonyl transcyclase, dehydratase, acyl reductase, ketoacyl reductase, and here, so these are the 7 and thioesteres. Okay, and here the ketoacyl protein is attached to acyl carrier protein, so it is attached to 4-phosphopantothiene. Okay, so only the dimer is functionally active, so the monomer cannot carry out fatty acid synthesis. Okay, so the fatty acid synthase functions as a single unit catalyzing all 7 reactions. Okay, so in that, so once the acetyl-CoA or malonyl-CoA enters the fatty acid synthase complex, so from that you are going to get, so it catalyzes all the 7 reactions, and the output is, it will form palmitic acid. Okay, so the two subunits lie in anti-parallel orientation. Okay, so the two subunits lie in anti-parallel orientation. So if you look at this, so here you can see, so the ketoacyl groups are in opposite direction. So here, so they lie in anti-parallel orientation. Okay. So the functional unit consists of half of each of subunit interacting with the complementary half of the other. Okay. So here if you look at this, so the functional unit consists of half of each of the subunit. So that is let me clear this. Okay, I'll show you. So here, so this is divided into two major types of units. This enzymes fatty acid synthase complex can be divided in two different ways. One is subunit division. In subunit division, so one whole monomer is considered to be one subunit. Okay, on the one side. So one monomer on one side is considered to be one subunit. Okay, so the functional division here, the functional division is, the functional division means, so the enzyme fatty acid synthase complex function by combining both these monomers, that is one half of the upper monomer and the other half on the same side of the upper monomer, that is other half of the lower monomer on the same side. So combining this will form 7 enzyme unit. So this is considered to be functional unit, that is, so this half part of the dimer will synthesize one molecule of fatty acid and the other half will catalyze the synthesis of other molecule of fatty acid. So this fatty acid synthase complex can be categorized into subunit division, where the upper part is considered to be one monomer and the lower part of the dimer is considered to be other monomer. So here dimer means it will have one half of the molecule will be identical to the other half. So here the dimer is in dimer, so the monomers are in the upper and lower part. So it is represented as upper part and lower part. So upper part is having one monomer and lower part is having the other monomer in this dimer. So it can be categorized into subunit division and functional division as we discussed. Okay. So the two functional subunits, that is functional subunits of fatty acid synthase, operate independently and synthesize two fatty acids simultaneously. Okay. What is the advantage of multi-enzyme complex? The intermediates of the enzyme reaction can easily interact with the active site of the enzyme. It need not search for its enzyme anywhere else. They are available all at one place. So here one gene codes all the enzymes. So the single gene will code for the biosynthesis of all these enzymes. So all enzymes are in equimolecular concentrations. So there will not be any deficiency of any one particular enzyme. If there is deficiency, so the deficiency will be the whole fatty acid enzyme synthase complex, fatty acid synthase complex. Okay. So hence the efficiency of the process is enhanced. Okay, so the reactions of fatty acid synthase complex. So now we will discuss about the reactions of fatty acid synthase complex. So this can be discussed in 3 domains. So the first domain is condensing unit, second domain is a reduction unit, and the third domain is a releasing unit. So the first domain, it is the initial substrate binding site. So here the enzymes involved are beta keto acyl synthase, or what we call it as condensing enzyme, acetyl transferase, and malonyl CoA ACP transcyclase. Okay, CE, AT, and MT. So the second domain is considered as is called as a reduction unit. So here it contains enzymes that reduce the molecule. So that is, it contains a dehydratase, anoyl reductase, and beta keto acyl reductase, and acyl carrier protein. Okay, so the acyl carrier protein is a polypeptide chain having a phosphopantothioene group to which acyl groups are attached in thioester linkage. Okay, so the ACP acts like an coenzyme A carrying fatty acyl groups. Okay, so acyl carrier protein will act like a coenzyme A carrying fatty acyl groups. Okay, so the third domain is considered to be releasing unit. So it is involved in the release of synthesized fatty acid in the cytoplasm. So it contains thioesterase or deacylase enzyme. Okay. So the reactions of fatty acid synthase complex. Okay. So the first step is, that is the 2-carbon acetyl CoA is transferred to acyl carrier protein of fatty acid synthase by acetyl CoA ACP transacylase. Okay, so here the acetyl CoA is transferred to acyl carrier protein, that is it is containing two units, that is ACP and cysteine residue. So where the acetyl CoA can bind, so acetyl CoA is transferred to ACP, that is acyl carrier protein of the fatty acid synthase complex. So this transfer is catalyzed by acetyl CoA transcyclase. Okay, so here COASH is released, so acetyl group is transferred to fatty acid synthase complex. So then this acetyl group is transferred to cysteine residue. So this acetyl group is transferred to cysteine residue. Okay, so in the next step, so the acetyl group is transferred to cysteine residue. So in the next step, so the acetyl enzyme so formed will combine with the molecule of malonyl CoA. So here the COASH is released, and malonate is transferred to ACPS unit of the fatty acid synthase complex. So this reaction is catalyzed by malonyl transacylase. Okay, so further, so this acetyl malonyl enzyme so formed will lose a molecule of carbon dioxide. So that is, this will undergo decarboxylation reaction. So by the action of beta ketoacyl synthase enzyme or condensing enzyme. So this will form beta ketoacyl ACP. So the next step is, so the acetyl group in the attached to cysteine residue is transferred to ACP group. Okay, so it will transfer to ACP group, ACP. So further, this beta ketoacyl ACP is reduced by NADPH dependent beta ketoacyl reductase. So this requires a molecule of NADPH that is converted to NADP, and so here the two hydrogens from NADPH2 are transferred to the beta ketoacyl ACP. So here one hydrogen is added. Okay, one hydrogen is transferred. Okay, so that will form beta hydroxy acyl ACP. It will form beta hydroxy acyl ACP. Further, this beta hydroxy acyl ACP undergoes dehydration. It will lose a molecule of water by the action of beta hydroxy acyl dehydratase. So this is an elimination reaction. It will lose a molecule of water from beta hydroxy, that is OH group, and the hydrogen from the neighboring carbon atom. So this hydrogen is removed. So it will be a double bond is created over here, and it will form trans-enoyl ACP. It will form an alkene trans-enoyl ACP. Okay. The trans-enoyl ACP, so that is formed, will undergo reduction by the action of enoyl reductase enzyme. So here the NADPH2 is contributes the reducing equivalent hydrogens. So the two hydrogens from NADPH2 adds to the carbons of containing double bonds. Okay, carbons containing double bond. So and it reduces the trans-enoyl ACP to butyryl ACP. Okay, enoyl ACP is converted to butyryl ACP. So now at this stage, the 4-carbon unit attached to ACP is a butyryl group. So it is attached to 4-carbon unit is attached, and this 4-carbon unit butyryl group is transferred from ACP unit to cysteine group. So it will form acyl S enzyme. Okay, so this acyl after this, So this acyl S enzyme, after the formation of acyl S enzyme, so this pathway is repeated. So that is the reactions of 2 to 6 are repeated 6 more times. So that is at the reaction 2, that is at the stage 2, malonyl CoA will enter this fatty acid synthesis pathway. So it gets attached to ACP. Again, it is transferred, and this 2 to 6 steps are repeated until you're going to get an 16 carbon unit fatty acid, that is palmitic acid or palmitate. Okay, so this palmitate or that is attached to fatty acid synthase enzyme is released by the action of palmitoyl thioesterase. Okay. So in summary, so this the reactions of fatty acid synthase can be discussed in 3 domains. That is, the first one is condensing unit. So here initially, so the substrate will bind to the fatty acid synthase unit. So that is acetyl CoA and malonyl CoA will transfer the carbon units. So the enzyme involved here in this reactions is beta ketoacyl synthase or condensing enzyme, acyl transferase, and malonyl CoA ACP transcyclase. In the second domain, it undergoes reduction reactions. So that is why, so the second domain enzymes are called as reduction units. So it contains dehydratase enzyme, enoyl reductase, beta ketoacyl reductase, and acyl carrier protein. So the acyl carrier protein is a polypeptide chain. It has an phosphopantothioin group, so to which the acyl groups are attached in thioester linkage. Okay, so to the ACP group, the fatty acid chain is attached. Okay, so the polypeptide, it is a polypeptide chain. So the acyl groups are attached to the ACP. ACP acts like an coenzyme A carrying fatty acyl group. Okay, so the third domain is a releasing unit. So it is involved in the release of synthesized fatty acid in the cytoplasm. So this is catalyzed by thioesterase enzyme or deacylase enzyme. Okay. So the total, to summarize, the end product is of fatty acid synthesis is 16 carbon unit palmitate saturated fatty acid. Okay. So in the first step, 2 carbon unit acetyl CoA will transfer the 2 carbons directly to fatty acid synthase complex, and further to it, in each cycle of fatty acid synthase reactions, 2 carbons are contributed by malonyl CoA. That is, in total of 8 cycles, that is in total for an 8 acetyl CoA units. So that is 14 carbons are contributed by malonyl CoA and 2 carbons are contributed by acetyl CoA. So in 7 cycles, so acetyl CoA is contribution of acetyl CoA is only in the first cycle of fatty acid synthase reaction, and in the first cycle malonyl CoA also contributes 2 carbon unit, and in the next 6th cycle only malonyl CoA will contribute the carbons. For each cycle it will contribute 2 carbons. So here in total, that is for an fatty acid which is having the capability to release 8 acetyl CoA units, so that is equal to 1 acetyl CoA and 7 malonyl CoA. So that is to say, so it is having contribution of 1 acetyl CoA molecule and contribution of carbon atoms from 7 malonyl CoA molecules. Okay, so this requires 7 ATPs. For synthesis of 1 palmitic acid, it requires 1 acetyl CoA molecule, 7 malonyl CoA molecules, 7 molecules of ATP, and 14 molecules of NADPH. Okay, and 6 water molecules. Okay, so this is the overall reaction. So this results in the synthesis of 16 carbon unit palmitoyl CoA. Okay. So further to this, palmitate is a precursor of saturated and unsaturated fatty acid. Okay. So further, different fatty acids can be formed by saturated, that is different saturated fatty acids which are having carbon number of carbon atoms greater than 16 carbons, so are synthesized by elongation system. And unsaturated fatty acid, that is this palmitate, can be converted into unsaturated fatty acid by desaturation mechanism. Okay, what is elongation of fatty acid? Okay, so elongation of fatty acid can occur by two ways, that is one by microsomal elongation system and another by mitochondrial elongation system. In microsomal elongation system, so that happens in endoplasmic reticulum of liver, and in mitochondrial elongation system it happens in mitochondria, and this is less active, and more number of times it is elongation process happens at endoplasmic reticulum of liver cells. And malonyl CoA is the donor of carbon units in elongation system again. In case of mitochondrial elongation system, it is acetyl CoA which is the donor of carbon units, that is 2 carbon units. It elongates fatty acid containing 10 carbons to 22 or 24 carbon. So these are considered to be long chain, that is microsomal elongation system synthesizes long chain fatty acid. So that is present in sphingomyelin. So that is important for myelination of nerve, which plays an important role in insulation of nervous system. So by mitochondrial elongation system, only medium chain fatty acid and small chain fatty acid can be synthesized. Okay, so this is a 20 carbon unit. So the first unit can be synthesized, that is the first 16 carbon unit is for palmityl CoA over here. So it can be synthesized by fatty acid synthase complex reactions, and further by elongation process it can be elongated up to 24 unit carbon containing fatty acids. Okay. So the unsaturated fatty acids, that is conversion of the saturated palmitic acid to unsaturated fatty acid, can be carried out by desaturation system. So this happens in smooth endoplasmic reticulum. So there are 4 fatty acyl desaturase enzymes in mammals. Okay, so they are designated as a delta 9, delta 6, and delta 5, and delta 4. That is, so what does this indicates? This indicates delta 4 means, so the double bond begins at the 4th carbon atom, and the double bond is in between 4th and 5th carbon atom. So the double bond is in between 4th and 5th carbon atom. Delta 5 means the double bond is in between 5th and 6th numbered carbon atom. Delta 6 means the double bond is in between 6th and 7th carbon atom. Delta 9 means the double bond is in between 9th and 10th carbon atom. Okay. So in mammals, there are 4 fatty acyl desaturase enzymes designated as delta 9, delta 6, delta 5, and delta 4. These enzymes will catalyze the desaturation process. But that is delta 9 desaturase enzyme will add double bond at carbons between 9 and 10. Delta 6 desaturase enzyme will add carbons between 6 and 7. A double bond between 6th and 7th carbon. Delta 5 desaturase enzyme will add double bond between 5th and 6th carbon atom. Similarly, delta 4 will add double bond. Delta 4 desaturase enzyme will add double bond between 4th and 5th carbon atom. Okay. Palmityl CoA is in 16 carbon containing unsaturated fatty acid, that is palmityl CoA is unsaturated fatty acid. So the unsaturated fatty acid is, that is from palmitic acid, so it can be converted to palmitolic acid. So it contains 16 carbon fatty acid, and it is containing only one unsaturation site at 9th carbon atom. So stearoyl CoA can be converted to oleic acid. So here also it contains one double bond at the 9th carbon atom. Okay. So one of the limitation of mammals is mammals cannot incorporate a double bond by desaturation system beyond delta 9. Okay, so that is beyond ninth and 10th carbon atom. So hence polyunsaturated fatty acid cannot be synthesized. It has by our body, by mammals. So it has to be supplied in the diet, but plant contain delta 12, delta 15 fatty acyl CoA desaturation enzyme. Hence, if we consume plants containing polyunsaturated fatty acid, we can get polyunsaturated fatty acid. Okay, in summary, to desaturate system, smooth endoplasmic desaturation occurs in smooth endoplasmic reticulum. So there are 4 fatty acyl desaturase enzymes in mammal, that is delta 9, delta 6, delta 5, and delta 4 desaturase enzymes. So palmitoyl CoA can be converted to palmitolic acid. Palmitolic acid contains double bond at the 9th carbon atom, that is between 9th and 10th carbon atom. So here you have the structure. So this is the first carbon, second, third, fourth, fifth, sixth, seventh, eighth, ninth, and 10th. So the double bond is in between 9th and 10th carbon atom. Okay, so it is an delta 9 fatty acid. So steroyl CoA, it is an 18 carbon containing, so it can be converted to oleic acid by the addition of double bond at delta 9. So here a delta 9 desaturase enzyme can convert palmitolic acid to palmitoyl CoA to palmitolic acid and steroyl CoA to oleic acid. So mammals cannot incorporate double bond beyond delta 9, that is PUFA cannot be synthesized, so it should be supplied in diet, and that is mainly from plant sources which can synthesize polyunsaturated fatty acid containing double bonds between 12th and 13th carbon atom and 15th and 16th carbon atom. Okay, so the next thing that we are supposed to discuss is regulation of fatty acid synthesis. Okay, so this here we can so there are 2 types of regulation. One is short term control, which starts rapidly, but it regulates or controls only for a short period of time. So there are 2 types, allosteric regulation and by covalent modification. And long term control, it is a slow process and it takes hours to manifest, but it is a permanent thing, that is it lasts a longer period of time. So this can be done by enzyme induction or by enzyme repression. Induction means activating the enzyme, repression means deactivating the enzyme. Okay, so the first thing is allosteric regulation. So this works by feedback mechanism. Okay, like for example, so if the glucose level is higher, the citrate level, so it promotes the formation of citrate. Okay, if the citrate level increases in the blood, so it promotes the activity of acetyl CoA carboxylase enzyme, which converts acetyl CoA to malonyl CoA. So this promotes the formation of palmitoyl CoA. Okay, if palmitoyl CoA concentration increases, this inhibits the formation of NADPH, that is, this inhibits HMP shunt pathway and malic enzyme activity, and also this also inhibits conversion of citrate to acetyl CoA, and palmitoyl CoA also blocks the activity of acetyl CoA carboxylase. Okay, so here the major rate limiting enzyme is acetyl CoA carboxylase, and the activation and deactivation mainly depends on the concentration of palmitoyl CoA. Okay, so and then one more method is covalent modification. Okay, one more method of regulation is covalent modification. So here insulin plays a very important role. Insulin, glucagon, and epinephrine. Insulin activates acetyl CoA carboxylase. Okay, hence it promotes by dephosphorylation of acetyl CoA phosphorus. Acetyl CoA carboxylase enzyme, in fact, it exists in 2 states. One is active state, which is in the dephosphorylated state, and another one is inactive state, so that is phosphorylated state. So phosphorylation indicates excess amounts of ATP is present. Okay, phosphorylation indicate excess amount of ATP is present. So when it is phosphorylated, acetyl-CoA carboxylase will be in the inactive state, and by dephosphorylation it can be activated. So this dephosphorylation process can be induced by the higher levels of insulin will promote the activity of protein phosphatase, which will convert the inactive acetyl-CoA carboxylase to active acetyl-CoA carboxylase by dephosphorylation. Okay, so this will promote the conversion of acetyl-CoA to malonyl-CoA, which can further undergo de novo fatty acid synthesis. Whereas increased levels of glucagon and epinephrine will deactivate the enzyme by phosphorylation of acetyl-CoA carboxylase. So this will inhibit the de novo fatty acid synthesis process. Okay. So this is by covalent modification, where the enzyme can be activated and inactivated. Long-term control mechanism. So here it involves a change in the gene expression, which controls the rate of synthesis of these enzymes. Okay, insulin induces the enzyme activity, that is acetyl-CoA carboxylase, whereas glucagon will repress or deactivate the acetyl-CoA carboxylase. High carbohydrate diet and low fat diet will induce fatty acid synthesis, that is, which it will increase, high carbohydrate diet will increase the level of insulin, and that will promote fatty acid synthesis. So high fat diet, or during starvation, or where our body fails to utilize insulin, or insulin secretion is very less due to failure of pancreas, what happens is, so there will be blockage of fatty acid synthesis. There will be blockage of fatty acid synthesis due to excessive activity of glucagon. Okay. So the summary of fatty acid synthesis. So the site is, that is, it happens in liver, adipose tissue, mammary gland during lactation. So the place is, that is, fatty acid synthesis happens in cytoplasm. So enzymes required are acetyl-CoA carboxylase and fatty acid synthase. So it contains a dimer of 7 enzymes in each monomer. Okay. So the primary substrate is acetyl-CoA. After contribution of acetyl-CoA, malonyl-CoA contributes. That is, one acetyl-CoA is enough for one synthesis of one palmitic acid. So 2 carbon units are contributed by acetyl-CoA. The remaining 14 carbon units are contributed by malonyl-CoA. In each cycle of fatty acid synthase complex reaction, one molecule of malonyl-CoA is contributed. Okay, and in overall synthesis of one molecule of palmitic acid, only one molecule of acetyl-CoA is contributed, and plus 7 molecules of malonyl-CoA will contribute to the carbons of palmitic acid. So the final product is palmitate, or what you call it as palmitic acid. Okay. So this is called as a Lennon's spiral, or a lipogenesis, the synthesis of fatty acids. So this is not a reversal of oxidation, that is beta oxidation. So beta oxidation is catabolism, and fatty acid synthesis anabolism. So we will look into the difference between beta oxidation and fatty acid synthesis. So beta oxidation occurs in mitochondria, and fatty acid synthesis occurs in cytoplasm. So the intermediate that are present as coenzyme A derivatives in beta oxidation, and the enzymes are present as independent proteins, and 2 carbon units are released as acetyl-CoA, and in each cycle of beta oxidation, coenzymes that are required are NAD and FAD for beta oxidation, and carnitine transport process works in beta oxidation for the transport from cytoplasm to mitochondrial membrane, transport of acetyl-CoA, that is fatty acid. So the end product is acetyl-CoA over here. So the intermediate, that is in fatty acid synthesis, occurs in cytoplasm. So the intermediate required is covalently linked to SH group of acyl carrier protein. So enzymes are present in the form of multi-enzyme complex. So malonyl CoA adds sequentially for each cycle, and the starting compound is acetyl CoA, of course. So the coenzyme is required is NADPH. So the transport system is a citrate transport system, and the end product is palmitate here in this case. So this is anabolism of fatty acid, and this is catabolism, that is, it is breakdown. Beta oxidation is breakdown, and fatty acid synthesis is uh, formation of fatty acid. Okay, so finally, so one suggestion is to tackle the zombie of procrastination. Procrastination means like postponing the things or finding a deficiency in each and every event of your life. So you can tackle by focusing on the process instead of product. So don't focus on the outcome, rather focus on process, like how it has to be done, what to be done to make it beautiful, how you can achieve the task, what should be the process, and work on that. Of course, the product will always be good and your achievements will be higher. Okay, happy learning to all of you. Thank you.